same thing is gonna be true for tonight's lab, which is 20, I think it's 21 that we're doing tonight. And, it's a mid-sagittal section, and this is the only way that you can see some of the, structures that are internal to the brain. What you saw in the other one were the convolutions, specific areas of functionality, and so just make certain that you're able to identify the structures and the areas on an unlabeled model. So you use the coded model, the color-coded model first, and then you transfer that information on over to the non-colored model so that you're able to identify, okay? Sutures, not sutures, I'm sorry, but fissures, parts of the brain, lobes, Broca's area, Wernicke's area, precentral gyrus, postcentral gyrus, association areas. Those are all the things that you need to be able to identify. So I would suggest that tonight that you go back and review the slide, okay? On the, it's not the medullated nerve, but it's the slide that has the multipolar neuron and the neuroglia. It says ox spinal cord or something like that, or ox, I'll double check it and I'll put it up on to the screen as well. But make certain that you do the review on that, okay? And I'm going to talk a little bit about the comprehensive exam. But I also am going to have to tell you that, frankly, I'm not going to be here next week. And I'm sorry about that. I have a family reunion that has been in planning phases and I got the wrong dates. I know. So the wrong dates then require me to be away, okay, as a result. So what I will do is I will probably, I'm accessible by email. So I'll be checking my emails every night. So if you have any questions on anything, we do have a sub. Professor Shaw is going to be coming in on Tuesday and Thursday, and he'll be giving you, going over then the spinal cord, and then I've asked him to also make certain to do a review with you. He's really good. He and I worked together way back when we started working together back in, some of you weren't even probably born, back in 76. Okay, got it. And then when we were both teaching over at La Sierra University at the same time, and then we both came over here at the same time. So we've been working together for a long time, and he has been typically on the schedule, but he got ill last year, so he pulled out, and he hasn't really committed to doing a term yet until he feels a little bit better. So I asked him to come on in because he's got the same style of teaching as I do. And so I met with him this morning and gave him all the information, all the outlines, the PowerPoints. I also gave him a group assignment that I want you to do next Thursday. So a week from today, you will have a group assignment. There will be 30 questions on it, okay? And I asked him to give you the group assignment in lecture so that the models are not around, but you sit together as a group to be able to answer the questions on there. And it will be over pretty much everything from labs numbers 17 through 21. Okay, so it will be it's focused in on lab information. Okay. So again, I'm accessible. I feel like I've never ever done this in my entire life. Okay, and so I feel like kind of a delinquent. But there's not much I can do about it. All right, so do you have any questions at all? Well, I'm going to go over the comprehensive just to make certain, because I want you to know how to study for this. I'm hoping that each of you took my recommendation and actually worked on the study guide for each one of the sections or each one of the units, and that then becomes your review. I would use that. I would not go back to read anything, because at that point in time, in terms of the comprehensive material, it's just a matter of being able to recall. So if you just go through and read the study guides that you've answered the questions for as a way to jog your memory, recall that term? Do you remember that concept? Do you remember the process? Okay, that will help you for this, for each of the study guides. Then I've kind of condensed it into this as well. So you can use another checklist. I would suggest that maybe you go ahead and look at the PowerPoints if you want to, but again, you've seen them, you got the information. My exam, when I come back, I'll go ahead and write it on up, and so I'll be using this and my study guides to help make my exam. All right, so let's go over the comprehensive portion. Now, if you'll notice here, I have 100 questions. I'm not positive, it could be 110. So I put down approximate because I'm not really certain, okay? It may be 50-50, I don't know, maybe 50-60, I don't know. But the bottom line is that it'll be about 110 questions, and it will be out of then 100. So you've got 10 points, so that kind of helps you for bonus, okay? Okay, so what I did was I went through the first 3 chapters, basically, Unit 1, and try to give a summary. I mean, I personally am not going to take a lot of information out of that unless it's applied, okay? So when we talk about the various ions that are responsible in action potential, that was discussed early in Chapter 2, but I'm not going to go back and review Chapter 2. It's only where it's applied, you know what I'm saying? And I'm not going to ask you, hey, define this term. No, because what I'm going to do is I'm going to say, true or false, the radius is lateral to the ulna. Okay? So that, you need to be able to apply the terminology rather than regurgitating it. Now it's the point where you've not incorporated. So, apply directional terms, that's what I mean there. Go back and review the abdominal pelvic regions because I need you to make certain that you know which region we have. What is the boundary that separates then the thoracic cavity from the abdominal cavity, right? And then where do we have then the pelvic cavity? Okay, so what's a dorsal cavity? What is a ventral cavity? So those are the different kinds of terms that you should be able to carry on in terms of like, okay, that I know that you're going to have to know for 50, okay, for 50B. Okay, so go back and review that. Now, pH is really critical for patient care. So you need to go back and just review what do we mean by pH, okay? And what happens when we have denaturation? Which is the acidic range, which is the basic range? Because again, the 50B is going to be really heavy on maintaining acid-base balance in terms of clinical diagnosis and assistance for patients. So go back and review that, okay? Again, bonds, how do we form those? Which bonds are easily dissociated in water? Which ones remain intact unless there's an enzyme that breaks that bond? And then if we have to use an enzyme, then what is an enzyme, basically? And substrate enzyme specificity is 1-to-1, okay? And typically we've got catabolic reactions, and then we've got anabolic reactions, and what's the difference between the two of those? So what do we need to make that ATP energy? And we've seen it a number of times. We saw it in I think it was chapter 3, maybe 2, with regard to just what's ATP, okay? And where is it produced? In the mitochondria. But then the actual utilization, like in the sliding filament theory, or when we are needing it when we're doing exercise physiology. So the applied is now important. It's just, you know, we already know that you know where it comes from. We know that you know the composition. But now how do we use that in the body, okay? And then it's real important to maintain the difference between organic versus inorganic compounds, so I think that that's pretty straightforward as well, okay? Which ones are covalent? Which ones are organic? And those are the ones that are building blocks for your cells and various structures within the cell. Okay, then the processes, and I've listed some. I'm just sitting there, you know, thinking, what is it that, in terms of the processes that you should remember? Okay, well, obviously, this is a review. You've had it in bio. You had it in chapter 3. So now we know what the organelles are. The organelles now are applied like Nissl body. What is that? It's a different name, but it has the same function as what we learned in chapter 3. And then, again, go back and review that phospholipid bilayer, composition of the cell membrane. And when I say cell membrane, we're talking about plasma lemma. So if we go to a myofiber, sarcolemma. If I go to a Schwann cell, neurolemma. If I go to an axon, axolemma. So the suffix lemma always is going to tell you we're looking at a cell membrane, and so the composition is going to be very similar. The functionality is going to be very similar. So it kind of, you know, it progresses through. Okay, and then we have a listing of active and passive transport mechanisms. What does that mean? And how is that useful? How does that apply? I do have to go back and ask about DNA replication, the term semi-conservative replication, and some of the enzymes associated with it, DNA polymerase, helicase, you know. So just a kind of a reminder, because your piece test is really heavy on all that. It's heavy on cell and molecular. Okay, so you need to go back and know DNA replication, protein synthesis, what's transcription, what's translation. So again, we'll list those for you. Go back and review mitosis. Cell cycle. Where's mitosis in terms of interphase? What's interphase? What do we mean by that? When does that happen? And what are the various phases there? Then instead of going through the histology per se, I'm going to start with an organ, such as skin, okay? Or an organ, such as bone, or an organ, such as muscle, and be able to then identify the principal tissue types that are within and what is their function. Now, the skin has, and I put that down because you need to go back and review some of the neuroreceptors, okay, the sensory receptors. So we know we've got some fine touch, we've got some for pressure, so you go back and review that as well. Go back and review burns, because that's, again, clinical. You're not going to see it again, okay, until you bring it perhaps up in the urinary system, where you have then an overload of proteins that go into the kidneys as a result of burn. When you burn the muscles, then that means that your kidneys are going to have to filter it, and it can become then overwhelming. So it can cause kidney damage, kidney failure as a result of having a burn. Okay, so go back and review the 3 basic layers. What do we find in those? Okay, and correlating it then to the burn, which layers are going to be affected? 1st degree burn, 2nd degree burn, 3rd degree burn. Okay. Okay, then go back, and what I do then is now that we've gone through multiple systems, and each system, in fact, even when we look at tissues, we've got specific cell types, right? So there's a lot of terms that are associated with cell types. So I've listed some of those, just sitting there kind of thinking, you know, off the top of my head here. Okay, so I've listed a lot of different cells. You should know the function of each one of those and where those are found. Okay. So cells are really, again, very, very important. Those are all listed for you. The skeletal system, in terms of axial skeleton versus appendicular skeleton, kinds of fractures that you're going to find, perhaps. Okay, so it's just a review of that composition, because it carries through. We're talking about tissue types, endosteum, periosteum. Why is that important? Okay, and then what's the organic component of bone? What's the inorganic component of bone? What minerals do I need for the inorganic component? What proteins do I need for the organic component? So it's just, again, now I am going to put in there parts, specific bones. So how many carpals do I have? How many metacarpals do I have in one hand? How many tarsals do I have in one foot? How many pairs of true ribs do I have? Okay, so I'm going to have a couple of questions that are associated with that. And I call it number play. Okay? So how many phalanges in one hand? So things like that. So go back and review that. Okay, and then I've got to incorporate some of the joints. Okay, what is the function of a joint? What, in terms of the overall skeleton, is the weakest point, right, where two bones articulate, and then the flexibility of movement, so diarthrotic joints. So we talk about synovial joints, or, because again, that's clinical. And when you have arthritis, rheumatoid arthritis, or osteoarthritis, what part of the body is being affected? And then just a sampling. I mean, I put this down. Do you know what the principal tissue types are again? You know, and then if I were to say bone, what's my principal tissue type? If I were to say ependymal cells, what's my principal tissue type? Now some of you might go, oh, that's nervous tissue. No, it's not. Okay. So you've got, now that it's part of that, doesn't mean that it's called that tissue. Now, if I were looking at a neuron, yeah, that's nervous tissue. If I'm looking at ependymal, oh, that's epithelial tissue. Right, so it's different. Even though it's in the nervous system, it's got a different origin. Okay, and then I put down a number of different physiological processes, action potentials, okay? If I talk about aerobic respiration, if I'm talking about transmission of nerve impulses. So again, these are things, just check off. Yeah, I know that. If I go excitation, contraction, coupling, what is that? I know that. What's sliding filament theory? I know that. What are the molecules that are associated with that? I know that. What kind of energy do I need? Do I need energy? Okay, so again, just a checklist for you. Oh yeah, I know that. Okay, and then just on muscles, I just want you to know major groups. So if I say the quads, if I said the hamstrings, if I said the rotator cuff, what are the muscles that are associated with that? And hopefully, again, it's clinical for those of you who are going into kinesiology, PT, OT, sports training, athletic training. You need to know that. So, and it's relevant to any of you who decide to go out and exercise. So we want to make certain that you're able to identify that. Now, I've listed a number of different components for the nervous system. This is the unit exam. Now, what I will do is on the exam, the final exam, which starts at 5 o'clock on that Thursday, right? It's June 6th. Okay, so we'll start at 5 o'clock, and you'll get 2 hours to finish it if you want to. You can take this, you know, doesn't really matter. And then I'll go ahead, and then that's it. No essay questions, no nothing. Now, the first part will be on the central nervous system. That one is going to be really detailed. Okay, so what you've seen in the lab manual, what you've seen on the PowerPoints and on the outline, you should know very well in terms of that. Okay, so you have then that information listed for you, terminology, neurophysiology. I've got everything on there as much as I can do it. Go back and review the lab manual because the lab information is, again, integrated into the lecture, right? So, two overlap very much, so it should be, and one's a visual, okay? Most people do well on the hands-on part, and then it's a little bit more difficult when it comes to then processing in terms of the lecture component. But that's your review. If you have any questions on anything, again, send me an email. I'll get back to you. So I'll be checking the emails each night. So if you've got anything, I won't get to it until the evening. Okay? Okay. Any questions on anything so far? Okay. All right. So now let's go ahead and go into the brain, part 2. I did the cerebrum. So all that information was on the cerebrum. In terms of functionality, locations, etc. Now we're going to go into the mid-sagittal section, and this is sometimes referred to, this part 2 is pretty much referred to as sometimes the primitive brain or the reptilian brain because it possesses all of the information for survival. The analytical thinking is on frontal lobe. It's not going to be in this area that we're going to be looking at, okay? So we're going to have then, I've already given you the various components of the, we've got the cerebrum, and then we have the core, within the core of the cerebral hemispheres, I've got these various structures, okay, or regions. So the first one we're gonna look at is the diencephalon. Let me turn on my little thing here. Okay, let's get this going. Okay. The nice thing about this is it's pretty straightforward, but you do have a lot of stuff to learn. Okay, so I want you to enjoy your Monday. Enjoy some time, but help this to you can enjoy the family and then balance. Okay. So let's look at the diencephalon. It's when you take your cerebral hemispheres and you separate them apart, as you look at the mid-sagittal view, then you'll notice that what you see is predominantly going to be the core, the diencephalon. So the diencephalon is going to form the wall of the third ventricle. Okay. So I've got my two lateral ventricles. Each one of those are going to go into my cerebral hemispheres. I've got an intervertebral foramen that takes the CSF and brings it down into the third ventricle. The third ventricle is a space that is formed when I put the two halves together. So it's not a concrete space, it just happens to be because of the way in which the diencephalon is formed. It then is kind of curved and it has a small space inside, and that's my third ventricle. So these structures are going to surround that third ventricle. The three parts to this is the core. The most important is the thalamus. Then you have the epithalamus, that means upon, and then you have the hypothalamus, that means below. So what you've got then is this clustering in the median aspect of the brain itself. So here we've got, this is what you're going to be doing tonight, the first part of the lab, is you're going to go ahead and label the various components. We'll make certain that you've got them all correct, so we'll go over it just as we always have. Toward the end of the lab, we'll make certain that your answers are coinciding with mine. And so you can see then that the diencephalon is all of this section that is identified right here. That's my diencephalon, okay? And you can see that in the roof of the third ventricle, that is going to be then a choroid plexus. The choroid plexus is a capillary bed that has also ependymal cells that's going to then form the CSF. So the question on the lecture test is, which of the following structures is important in the production of cerebral spinal fluid? Okay, and again, every ventricle has one of the choroid plexus in it. Even in the two lateral ventricles, you have a choroid plexus. So here you have then the diencephalon. Okay, now a couple of things about the diencephalon. This is where it's now been highlighted, okay? And you can see here, this is a cross section through my, this portion here, right here, is my hypothalamus. This large area is my thalamus. And then between my thalamus, because again, I need to have communication between the two halves, okay? And so those are called commissural fibers, okay? So they're white fibers that allow for communication. So in between the two thalamus, I'm going to have this. Now, it's given a lot of different names. So again, I know the multitude, so interthalamic connection, interthalamic mass. And here it's called the interthalamic adhesion. I don't really care which one you put it on. Whichever one you want to go ahead and name it. I know the different names that are associated with that. So this allows for then the communication between the two thalamus. I don't want the right side of the body working independently from the left. It's coordinated, okay? And so I have between this large structure here is my thalamus, and it is a nucleus. So what do we find predominantly in that area? Do I have myelinated axons, dendrites, or do I have a nucleus
within the CNS. So what is the greatest abundance of structures? Okay. It's gonna be right now, it's gonna be neurons, right? Cell bodies of the neurons. That's what we're gonna find predominantly in here. Okay, the intrathalamic connection is the actual axonated, they are myelinated axons that are gonna connect between the two. Okay, and then down below, if this is my thalamus, then down below, that's gotta be my hypothalamus. Okay, so there's my hypothalamus. And then my epithalamus is this over here, on the posterior aspect. So on the anterior inferior view, I'm gonna find my hypothalamus. And then, I'm going to find on the posterior view, I'm going to see my epithalamus. So if I were to look at this on my mid-sagittal section, this happens to be the optic chiasma. Now, do you know where that optic chiasma would be? It's the two optic nerves coming from the eyes, from the retina, that are gonna crisscross and X and go to the opposite sides of the brain. Okay? So the X is a chiasma. It sits right on top of, your anterior clinoid process and the shelf that connects the two, your optic chiasma sits right on that. Okay? And then coming down from that hypothalamus, this structure here is the pituitary, or the hypophysis. And we already know where that's situated. That's gonna be found in the sella turcica, okay, within the hypophyseal fossa, okay? Okay, so there's my pituitary. And then here, I'm gonna have in the back, that's my pineal. So on the epithalamus, it's gonna be the pineal. So on the lab test, you need to be able to identify these structures on the mid-sagittal section. So on here, just to kind of back up, here's my corpus callosum. Okay, does anybody recall what that corpus callosum is? It's the connection between the two cerebral hemispheres, right? So that is made up of myelinated axons that communicate on either side. So they're called commissural fibers. Things that go across from one side to the other are commissural fibers. Just like your intrathalamic connection or intrathalamic mass, that is a commissural fiber. So here I find, right here, that's my pineal. And technically it's in the region called the epithalamus. But we also find in that epithalamus this posterior commissure. So the posterior commissure is right next to the pineal. And we'll talk about what those do in a moment, just identifying where this is at. If it's a posterior commissure, what does it do? Commissural tracts, right? So it allows for communication in the epithalamus, on the two halves of the epithalamus. I've got the connection between the two thalamus, intrathalamic connection, okay? And then if I've got a posterior commissure, well, I'm gonna have to have an anterior commissure, okay? So we're gonna find an anterior commissure. There it is right here. It's in the front. Okay, so it's on the anterior aspect of that diencephalon. And if it's a commissure, it means it's going to allow for communication between both halves of the diencephalon, but on the on the anterior aspect. Okay? Okay, so now let's go through, now that we've identified them, makes it easy to then label your diagrams. Now we're gonna go through some of the functions associated with it. So functions that become a little bit more difficult, I mean, we can find these things, identify them. Gotta be able to pull them up out of memory because it will be on the lab test. But it's the function that's gonna be included then in my lecture test, right? Okay, so the epithalamus is gonna be on the dorsalmost portion of my thalamus. It contains the pineal, and don't worry about the habenular whatever, nuclei, okay? So there are nuclei there. Obviously, the thalamus is all going to be gray matter anyway. So that's going to then be cytons of my cell bodies, of my neurons. Okay, it's, so the epithalamus, the pineal, is the source of melatonin. So it's going to help regulate the sleep-wake cycle, okay, kind of. Because we do have another structure that's gonna also be important in that. It's part of the circadian rhythm, so that it's not sleep-wake, but dark light, okay? So we've got then the melatonin. And some people take the melatonin because they feel that that's going to help them sleep much better, okay? It's soothing, okay? So you can take exogenous tablets of melatonin. The other thing is that tryptophan is an amino acid that makes you kind of sleepy as well. So after a Thanksgiving meal, when you're heavy loaded on turkey, okay, all my poultry has high levels of tryptophan. So not only did you overeat, that makes you kind of groggy because all the blood's going from the brain down to the stomach to digest what you've just eaten, but in addition, the tryptophan is in there. And another source of tryptophan is like in milk, whole milk. So when you drink warm milk before you go to sleep, then that also is going to be somewhat of a sedative, okay, because it's tryptophan rich, okay? So you can do it either way. Okay, now the thalamus, notice on the diagram, all of these different colors represent masses within the thalamus, and then within that mass, I've got a number of different nuclei. So it's really abundant in gray matter. It's got a lot of information relay that's coming through that thalamus. Okay, so this is where you can see it on the slide, like these slides, they're really very good. And then you can see it, if you're looking from a lateral view, where you might be able to find it, but this is actually then the various portions of that thalamus. Okay, so let's go through some of the functions associated with the thalamus. So on here you can see it's indicating that it is paired, and it's going to form the wall of the third ventricle. Okay, and then you've got a number of different nuclei that are associated with the thalamus, okay. And these nuclei are going to help register and synapse sensory information. In other words, all of my sensory information that's coming from my sensory receptors has got to be brought to the thalamus for interpretation. So all of my sensory, except for olfaction, that's the only one. The sense of smell does not do that, but everything else, taste, touch, vision, all of that's got to be processed through the thalamus to then go to the appropriate area of the cerebrum. So I'm going to have a synapse within the thalamus for that sensory input. Okay, so as you can see, except olfaction here. So you're going to have then the sensory area. Now the memory is also associated with the thalamus. So you have then sometimes a strong association of a particular sense with a memory. Like I was saying, the smell of turkey always brings up maybe Thanksgiving or some holiday, okay? So it has a memory connection. I'll tell you, I had a boyfriend in high school, and he used to wear all the time that Old English leather cologne. I could smell that, and all of a sudden it takes me back to high school. I can tell. It just goes right back. You know what I'm saying? So some of those different kinds of memories are very strong. Okay? And that's because they're laid down as a memory tracks along with and aligned with that particular sensory perception. Okay, so the thalamus is also implicated clinically in alertness. So whether you are over alert, in which case you might have ADHD, okay, that all originates from the thalamus. How that sensory information is coming in, and then how then it's going to be interpreted with the brain or how quickly it's gonna respond. So consciousness and alertness is associated with the thalamus. So can you see how I can make that into a multiple choice question? Which of the following areas of the brain is associated with the alertness, okay, in terms of consciousness? If you have damage to the thalamus, because everything that sensory goes there, you may have some psychological disorders as a result of damage to that thalamus. So one would be bipolar. Okay, that also triggers then the transmission of neurotransmitters, okay, or the release of neurotransmitters. In addition, you can see right here in this section, they've compared a normal coronal section of a brain. You can see again the gray cortex, that's the region. What do we find in there? Cytons of the multipolar neurons. Then the white matter, okay, what do we find in there? Myelinated axons predominantly, but we also have some unmyelinated ones in there as well. And you can then see the openings. This opening represents a portion of the lateral ventricle. I do have in the plexiglass in the lab, and I'll put it up on the monitor tonight, a section, coronal section of the brain. You can see then the ventricles. You can actually see the space. Okay, so there's an open space. That space then is filled in with cerebral spinal fluid. But if you notice, this is a normal healthy brain, and this is one where we have advanced Alzheimer's. So most of the variation is going to occur at the thalamus. So the ability to make that memory association, okay, like as you get progressively worse in the Alzheimer's, you can't recognize your own relative or your own daughter, your own son, because there's a disconnect. Now, where does that happen? In the thalamus, because it's associated with that memory tract, okay? So, we have then kind of a way to kind of help a visual. It's pretty dramatic. Pretty dramatic loss. And to be honest, I mean, I know this is kind of TMI, but one of the reasons why I continue to teach is because I don't want my brain to go like that. I figure the more I use it, the more I hydrate, the more healthy I am, the better, and that I'm going to keep that at bay. That's psychologically what I convince myself. Okay. Okay. So now let's go from the thalamus to then the hypothalamus. So the hypothalamus, you can see, is identified in the orange there. Now let's go through and be able to identify. Now this is looking at the brain from an inferior view. So sometimes what you have to do is you got to stop and go, where in the heck is that? Well, I don't get it. And you see then, there's my optic chiasma right here. So this happens to be the optic nerve that's coming from my retina of my eye, and then it's going to crisscross and go this way to my occipital lobe. Okay? Because my visual center, my visual cortex is on the occipital lobe. So it's going to crisscross at the optic chiasma. This optic nerve is going to pass through the optic canal. So if you've got a visual of the optic canal, you can see how it would pass it through, and then it's going to crisscross and then go back. Okay. Now you're going to have some fibers that are going to go. It's not all 100% that are going to crisscross. You're going to have about 20% that stay on one side and 20% on the other side. Okay. And they go online on their own side, but then the majority are going to crisscross. So what we're going to find is this little opening right here is where my stalk of my pituitary would attach. An inferior view of that. Then if you just kind of looking at this, if I go, this is going anterior. Okay. When you're looking at the models, before you start identifying, first determine, am I looking at the anterior side of the cerebrum or the posterior side? Because you're going to get two different answers, right? So you want to orient before you answer. And so what you'll find is that posterior to this area is these two small little projections. That's called my mammillary body. I'm gonna come back to that, okay? But those are my mammillary bodies, you know. This is the one that, actually, you know, when you go into a room and you go, and somebody has just vomited. Okay? It was just, you know, you just kind of walk in and you go, oh my gosh, and then sometimes it's so bad that you almost feel like you're going to get a gag reflex as a result. You might have that with, or even with an anaerobic infection. You walk into a room with a patient who's got an anaerobic infection, you're going, oh my gosh. And you then have a gag reflex that's associated with it. That's because the mammillary bodies have a heightened sense of awareness for the sense of smell. So the portions of that are going to then be activated. So as you can see, the hypothalamus is going to regulate the endocrine system. So almost your entire endocrine system is going to be stimulated or inhibited by the hypothalamus sending out chemical messengers to the pituitary. So they used to call the pituitary the master gland, and, you know, because they thought, oh, all of the important hormones are coming from that pituitary. However, the releasing factors that stimulate the release, or the inhibiting factors that inhibit the release, all originate from the hypothalamus. So it's actually the super control center for all of your endocrine system. So functions of the hypothalamus, and you can see there are quite a number of nuclei that are associated with it. So you might ask yourself, do we have to know all of these? How many did I put down in your outline? Did I put any of those in your outline? Okay. If it's in your outline, that's your guide. Right? So the preoptic nucleus regulates body temperature. So that portion of the hypothalamus is going to be affected when I take aspirin or Motrin or anything that's going to be a fever reducer. It's going to target then this portion to help regulate the body temperature so it's not too high. And then that also maintains homeostasis of your overall body temperature. Okay? Then we have an area which is going to be important in stimulating thirst. So you'll have this center is going to send out messages to your pituitary that says you need to release a hormone in order to create thirst, so that you're going to respond to that stimulus. Oxytocin is another one. Okay, that oxytocin, does anybody recall? This was way back when. Is this hormone a positive or a negative stimulus or feedback loop? We had two positive feedback loops that we gave as an example. And that was in chapter 1, if you can believe it. That's why you're going, what the heck is she talking about? Okay, negative feedback is that there's a mechanism to control it and turn it off. We negatively feedback. Positive is that once we get it started, we want it to go even more and more and more. Oxytocin is important for uterine contraction during labor and delivery. So once I get that thing going, I want to keep doing it. So sometimes what you do is if the patient doesn't have sufficient oxytocin, you give them a Pitocin drip, right? And that's going to then simulate even more oxytocin. And that's going to force, then that's a positive feedback because you want that uterus to push out that baby. That's positive feedback. The other one that most everybody can recall is the clotting mechanism. We start one clot, we want to form more clots. We want to plug up the broken blood vessel, right? That's a positive feedback loop. Okay, so I just wanted to bring up the oxytocin. So that's where does it come from? Oh, it comes from that nucleus, paraventricular nucleus in the hypothalamus. All right, so you can see then, this one here I want to talk a little bit about circadian rhythm. Circadian rhythm is like light, dark cycle, okay? It's kind of correlated to awake sleep. But melatonin is the one that we assign to sleep and wake cycles. If it's circadian, that means dark light. So what happens is that if you were hired to work the graveyard shift, right? And you're working then a 12-hour or 7-hour, whatever it might be, and you go in at 11 and you get off at 7 or 8. Okay, so what's gonna happen is that we usually sleep when it's dark. We usually stay awake when it's light. When you're working those hours, you are actually reversing it, right? You're making your body be alert during the dark, and then you're forcing it to go to sleep in the light. And now, you can do that for a while, and your body will get used to it, but after a while, about 5 years, 6 years into that kind of a schedule, you'll find that people get very depressed, okay? Because it's part of that thalamus, the diencephalon, where alertness, depression, it's all integrated. So what you'll find is that nurses who work those hours usually have to take some kind of antidepressant to keep them from feeling too depressed, because your body is responding. But you're still working that. You might be getting a step up, in terms of pay, whatever you might want to be doing, or maybe you were stuck there because that was the opening at the time. Most people want to work, the light cycle, you know what I'm saying? But circadian rhythm is really important too, because that also is going to affect your overall alertness. You've got to have a certain number of hours of sleep, for example, okay? And then, again, mammillary bodies. This one here is the one I was talking about. It's a relay station for olfaction. So when you walk into that room and you smell whatever you smell, and it's, how can I, well, you know what I'm talking about. Then it's gonna cause a gag reflex. Okay, it could potentially initiate a gag reflex. Okay, now, that's the diencephalon. So, if I were to say on a test question, which of the following is not part of the diencephalon? Corpus callosum, okay, third ventricle, thalamus, intermediate mass, or the pituitary? What is not part of that list I gave you? What's not part of the diencephalon? And corpus callosum. Corpus callosum is cerebral. Okay, it's the cerebrum. Okay, so now we know what the structures are. You're going to be labeling those on the diagram. And now we have an idea of what the functions will be. Now, for right now, in the 50A, we're just identifying brief functions. Then when you get to the 50B, we're going to start integrating and giving a bit more detail. So you'll, over the summer, at least before classes start up again in the fall for the 50B, you want to take about a week to just kind of review so that you don't have to go back and relearn. The assumption is that you already know it. So you want to be able to review, be able to do that. Okay, so let's now go from the diencephalon to the brainstem. The brainstem is all of this that's identified in the orange, okay? And so let's go through the portions of the brainstem. It's got the midbrain. It almost looks like it connects the cerebrum to the lower part, right? The lower centers, okay? Then we have the pons, which is, here's my midbrain. This is my pons, as you can tell here on the orange, and then the medulla oblongata. You don't have to write medulla oblongata for the lab test. If you just write medulla, we're okay with that, okay? All right, so the brainstem is going to control all of your autonomic behaviors, okay? So if it's autonomic, that would then be part of your CNS. Okay? The nerves that come away from that are going to be PNS. So the core is CNS, and then anything that extends away or brings information in is going to be part of the PNS. So we're going to find in this brainstem also fiber tracks information that's coming up into the spinal cord and going up to the cerebrum. They're gonna be in the white matter, in the spinal cord until we get to this area here. Okay, that's what this means by fiber tracks, connecting higher and lower neural centers. Okay, so let's look at first the midbrain, and I just call it the midbrain. And because it's my test, that's all I'm gonna do. But I used to, a long time ago, be able to ask students which portion of embryonic development would this be. So it would be then the mesencephalon, mes, met, mile. So mes is the midbrain. Met encephalon is gonna be my pons, and then the myelencephalon is my medulla. You're gonna learn about that in the 50B when you talk about your development, embryonic development of the brain. Okay, just call it the midbrain. We're okay with that. Okay, makes it easier. Okay, so the midbrain is composed mostly of tracts of nerve fibers. So if I say tracts of nerve fibers, what am I talking about? Which one? Okay, so the tracts are in the CNS, right? And they're myelinated. Right? Okay, white matter. And so what we have then, this is what it's gonna look like. There's my thalamus, just to get an orientation. Then this portion here is all of the midbrain. It reminds me of, as strange as this is gonna sound, it reminds me of like a broccoli. You know how you've got the flowerlets, and that would be over here. All the little things that are that are sticking out the green. And then you know how you've got the stalks that comes together in the broccoli? That then becomes my midbrain. Okay. So it then, and if you cut through the broccoli, don't you see it's kind of white? I try anything to kind of remember things. Makes it easier. Okay, so what does this midbrain do? They're going to refine motor movements. So when you are walking and you have a certain stride and you've got your legs going in one direction, your arms going in the opposite side moving, then that's kind of like coordination of that motor movement, and that's accomplished by way of that midbrain. So the fibers are going up, and that allows for that. In the posterior part, we've got this structure. This is now looking at a posterior view, and I got a model that shows this. I've got these two upper projections, two lower projections, and they're like two pairs, okay? And that group of four is called my corpora quadrigemina. So think about it this way. A corpora is a body, quad, four, Gemini, twin. So I've got a superior and an inferior colliculus, okay? That makes then four. So what I'm gonna find there, my superior colliculi are gonna be involved in peripheral vision, in the movement of the eye, okay? That acute sense of the periphery. And then if I'm looking at my inferior, the inferior helps me to determine where's the sound coming from. So when I hear a sound, don't you turn toward the sound? Where does that, I can hear it, but where does that integration and analysis of where that sound's coming from? That's gonna be my inferior colliculus. When I see something out of the corner of my eye and I turn my head that way, okay, that's my superior colliculus. So it's an acute sense of vision, usually peripheral. For the superior colliculi, just think about your eyes above your ears, okay? So that, eyes, and then hearing. All right, so that's my midbrain. You'll be able to identify those on the model. Now we're gonna go to the pons. So if I go from midbrain, then I go down to the pons, so that's inferiorly directed. And this is where I'm gonna find my fourth ventricle. So here's my pons, there's my cerebellum, and then between those two I have a space. That space is my fourth ventricle. So the question on the lecture test will be, where would you find a lateral ventricle? where would you find a third ventricle, and it will list the various areas, and then where would you find the fourth ventricle? So the fourth ventricle is between the pons and the cerebellum, and you'll see that on the model. And this area is critical because it helps to regulate your breathing depth and pattern. So your rate of breathing, okay, whether you're breathing deeply or if it's shallow breathing, that's all going to be determined at the pons. And you've got nerves that you're going to be learning, cranial nerves that come off of that then are going to innervate the diaphragm and various structures that aid in breathing. Okay, so you're going to make that connection later. The next part is my medulla oblongata. See, for this part here, it's just pretty much identifying on the lab. Can you name that and give me one function associated with it? Because you can choose any which one for that lab test. This end is my medulla oblongata. And this one here has my cardiovascular center. It's going to regulate the force and rate of heart contraction and also regulates vasodilation. Okay, so that's my medulla in that area. In addition, it works in conjunction with my previous in order to get my respiratory center. Okay, and then we've got other centers associated with it, so the way that the reason you cough with hiccups or whatever, you're going to have then the medulla that's responsible for that. Now the medulla has a number of cranial nerves that come off of it and then make their way to the various regions of the body. Okay, cerebellum. This one's easy to identify. Okay. Cerebellum, as you can see, these illustrate a number of different views of the cerebellum. When you see it on the model, there's no question. You know, that's the cerebellum. Contains about 11% of the brain mass. Okay, and what it will do is it takes the information from the cortex, the brainstem, and sensory receptors to provide coordinated actions of the skeletal muscle. So when you don't have enough dopamine and you've got Parkinson's, right, then that's where you have then a lack of coordination as you're walking too. So, wow. It plays a major role in balance, but I'm going to tell you that the majority of the sensory receptors for balance are located in the inner ear, but then it goes down to the cerebellum for balance. Okay, that's where it's going to be interpreted. Now, a couple of things that you're going to find on the cerebellum. Go ahead and give you the names first, and then we'll go ahead and show them one more time. It's connected because there's two cerebellar hemispheres connected by way of a vermis. Okay, so that connection, that would be an association, not an association, that's a commissural connection. Okay, is my vermis. And then you'll notice that on that cerebellum, it's got multiple smaller folds than what you would see on the cerebrum. We saw the convolutions on the cerebrum, the gyri and the sulci, occasional fissures here and there. But on my cerebellum, they're much smaller and they're deeper. Okay, so we call those then the folia. And then if I were to do a mid-sagittal section through my vermis, I would see the portions of white matter, and we call that the arbor vitae, or the tree of life. So let's go ahead and look to see what that would be. So here is my two cerebellar hemispheres, okay? And the connection between the two, you can see that little area, that's my vermis. If you look at the external features of the cerebellum, you see a number of small folds. Those are called my folia. Okay, so you're looking at it, you see all the folia that is associated with that. And this is what you see on the vermis right here. And then this is superficially the folia. And then if you look at this, you'll notice that there's white matter that goes up. You can see it better here. You can see then it looks like a tree trunk with multiple branches. okay, that extend out. That's why it's called, that white matter is called the arbor vitae, or the tree of life. So the arbor vitae is actually white matter. The folia are the external folds that are associated with the cerebellum. The vermis is the connection between the two cerebellar hemispheres. So the question, very easy, which of the following structures is found connecting the two cerebellar hemispheres? I'll have folia in there, and I'll have arbor vitae in there, I'll have vermis in there, so you're able to see the association. Now, we're gonna go into just a little bit on the limbic system, because the limbic system is where it processes our emotions. So all the structures that we looked at so far as part of the diencephalon, midbrain, etc., those are all structural for breathing, cardiac centers, etc. Just, that's why we call it reptilian or primitive brain, because it just maintains the basic functions of everything, okay? Regulates everything. But associated with humans are a number of various emotions. So we're gonna have then relays to the limbic system, which is then going to be interpreted and influenced in our behaviors, okay? It's gonna then allow for that emotional component. So like I said, Old English letter, sensory, goes up to my thalamus. Then it's gonna go into my limbic, and I go, oh yeah, I remember that. Those were good days in high school. Funny, really. Okay, so we've got then a number of different structures that are associated with the limbic system. Okay, and this is then, again, shows just the outline. We have the amygdala, and this is where we're gonna have then anger, fear, anxiety, and violent behavior, okay? So, again, this is where I'm gonna store some emotional memories. So, if I have any trauma, tragedies that happen in my life, they're gonna be registered here in my amygdala. They're gonna be filed there. The hippocampus, okay, is this portion here, and that campus is gonna convert short-term into long-term memory. So when we have short-term memory, we're learning something, we're processing it, and we have then a number of various sensory receptors that are going to be stimulated to help me in my learning. I gotta be able to read it. If I'm writing it, so I've got a number of different receptors that are gonna then be stimulated. And then what it's gonna do is go into my thalamus, and then I have to then sleep on it to get it into somewhat short-term memory so I can pass a test. But for long-term memory, I have to take it to the next step, which is then to the hippocampus. So if I'm studying it for maybe a test that maybe is a year later, that's long-term memory. So we do have short-term memory, you know how that is. You learn it really quickly, and then it's for short-term, and then you forget it the next day after you take your test. You go, okay. Because you didn't really take the time to process it completely. In order for long-term memory to occur, you have to have it going into a file that's in the hippocampus. Okay, so that's the long-term memory. Even the memories, the long-term memories would be things that you did when you were a child. Those are long-term memories, right? Okay, so the fornix is this portion. Let's see. Is a tract that connects the hippocampus to the hypothalamus, so that I have then an association processing memory. And then the cingulate, you're able to identify that on the model. The limbic system you can't find. It's diagrammatic, okay, because it's internal, it's lateral to the diencephalon area. It's deep to the cerebrum. But when I get into the cingulate, I can see that's actually part of my cerebrum. So if I have a mid-sagittal cut, you'll be able to see the corpus callosum on the mid-sagittal cut, and then just above it, I have this cingulate gyrus, just superior to that, And that then is gonna be for communication between the hippocampus and the thalamus. So can I find any part of this in terms of lab test? The only thing is a singular gyrus, you can find that. The other ones, you just need to know the name and what they're associated with. They're mostly associated with emotions and memory processing. It's another diagram that illustrates it. This is the cingulate gyrus, which is the corpus callosum is just inferior to it. Those are going to be commissural fibers that connect the two halves of the cerebrum. Then just above it, the gyrus just above, then we would find my cingulate gyrus. Okay, so you should be able to find on the models a precentral gyrus, a postcentral gyrus, a cingulate gyrus. Those are the ones at least we asked you to identify. Okay. So again, in order to get yourself oriented and be able to name it a precentral or postcentral, it's not very clear, is it? On some of those unlabeled brain models, they're not. I don't think that they're very good. They're very short. So get yourself oriented, and then the gyrus just in front of that central sulcus is going to be then my precentral, and then the postcentral behind, right? Posterior to it. Okay, all right, so that's it. Okay, so we'll go ahead and tonight's lab is lab number 20.